Hey guys, I feel like I haven't done a video in a while, and so we are now starting our last unit of the semester, which is going to be DNA and protein synthesis. And we'll be wrapping up our fall semester, getting ready for semester exam before too long. And so this is going to be our first lesson on DNA. Now the good news is I feel like a lot of this is going to be a review. In fact, when we did macromolecules in the beginning of the year, we definitely covered some of the basics of DNA and RNA and proteins, and so hopefully this unit will feel a little bit familiar to you. We are going to add some things that maybe you haven't learned before. Now, just to kind of give you a little bit of background, I know if you have your notes in front of you, you can see that the history is not on there. Yesterday you watched the video, the double helix, and it kind of showed you how the discovery of the DNA structure came about. We had kind of the race between several different people, with Watson and Crick kind of being the ones to actually build the first model. And so just to give you a little bit of history, you don't have to write this in your notes, but in your study guide there is a place for a timeline. If you wanted to get that out, you could jot some stuff down there, but you could always go back and do this later. But just to kind of give you some, I guess, context about this particular material. Just kind of, we just finished up genetics, and Mendel's paper was published in 1866. He actually didn't get credit for his work while he was still alive. It was after he had died. But his paper on genetics was published in 1866. So we did not have the model of DNA by Watson and Crick until the 1950s. So this goes to show you that a lot of what Mendel did, he did without the knowledge of genetic material. The concept of a gene didn't exist. The knowledge that DNA was genetic material didn't exist, so it makes Mendel's work that much more impressive. Now, the actual substance of DNA was discovered by the 1900s, and it was discovered that there were proteins and DNA in the nucleus, and so a guy named Frederick Miescher named it nuclein, and so this was the first discovery of the DNA. And so it took a while for us to know about the structure. Now, before we even knew that it was the structure, it had to be determined that DNA was the genetic material. And as you saw in the double helix video, a lot of people were leaning toward proteins. And so it took several experiments to conclude that DNA was genetic material and not protein. So these first few experiments that we're going to look at were the ones that kind of helped to determine that DNA was the genetic material. Now, Frederick Griffith, he actually didn't do anything with DNA specifically, but he did what we call the transformation experiment. So he was doing some tests with bacteria strains and mice. And so hopefully you're familiar with the concept that there can be different strains of bacteria, like for instance E. coli. We naturally have E. coli that live in our gut and are healthy for us, but then there's also E. coli strains that can cause us disease like food poisoning. So this is kind of what we had going on with Griffith's experiment. So he was working with a pneumonia bacteria, and one strain was non-virulent, which means it was not disease-causing, and then another strain was virulent, which means it does cause pneumonia. And so what he did was he put the non-virulent, the harmless bacteria into the mice, and they were fine, as you can see here. He put the disease-causing bacteria in the mice, and as you can see, the mice got pneumonia and died. So then what he did was he took the bad one, he took this virulent strain, and he heated it up to high temperatures and killed it. And so he put the dead, bad bacteria into the mouse, and the mouse was fine. And so the final experiment was he took some live of these, he took some live of the non-virulent, and he took the dead ones, and he mixed them together, okay, the dead, bad ones. And as you can see that by themselves, the non-virulent, the mice were fine. The dead, virulent, the mice were fine, but somehow when he mixed them together, the mice got pneumonia and died. And so this kind of led to what we know as transformation. And we're actually going to learn about transformation when we get to DNA technology, but essentially transformation is the uptake of foreign DNA. And so all Griffith concluded was that somehow something from those dead virulent bacteria transformed the live non-virulent bacteria into the disease-causing kind. Now, once again, nothing to do with DNA yet, but we're going to see why this experiment is important, okay? So all he did was say that transformation occurred. Something transformed those live bacteria. Now, the next group, Oswald Avery, and I believe he was mentioned in the video, but Oswald Avery and his group, they concluded that DNA was the transforming agent. So what they did was they repeated Griffith's experiments, but they tried to determine which macromolecule was responsible for transformation. And so they would break down the proteins, they would break down the DNA, they would break down, the lipids. And what they found was that it was only when they broke down DNA that the transformation did not happen. And therefore they concluded that DNA was the transforming agent. So this is kind of our first experiment that kind of points to DNA as the genetic material. Now this was further supported, okay, you can see several years passed, but this was further supported by an experiment done by Hershey and Chase. And this is another one that supports DNA as genetic material. And they did what's called the bacteriophage experiment. I want to say sometimes it's called the blender experiment, but this is kind of what they did. So they tagged proteins and DNA with radioisotopes. All right, so the use of radioisotopes allows them to kind of visually see these molecules. And so they used radioactive sulfur in the proteins because proteins contain sulfur. And they used radioactive phosphate in the genetic material. So what they were looking for is they had these little bacteriophages here. And so bacteriophages are viruses that infect bacteria. And they kind of have this, I like to call them cookie robots, but they kind of have this protein shell that kind of has this head and tail type structure. And then inside is the genetic material or the DNA. Now, what they were trying to figure out is they knew that viruses had to get inside of a cell and kind of take it over, but they weren't sure which material they were using to do that with. They didn't know if they were using proteins to take over the cell or if they were using the DNA to take over the cell. And so they tagged the protein. You can see that the protein appears pink here, so that is because of the radioisotope. And in here you can see that they tagged the DNA and it's blue. And so what they were looking for is which one of the materials is used inside of the bacteria cell to create new viruses, because that would indicate that it was the genetic material. And what they found was it was the DNA. So we can see here that they were able to see that radioactive DNA inside of that bacteria. And so once again, we conclude that DNA is the genetic material. Now, once it is concluded that it's most likely DNA as the genetic material, as you saw in the video, there were still some people that didn't fully buy in just yet. And so we kind of start to have this race in the 1950s to determine the structure of DNA. And so several of these people, if not all of these people, were mentioned in the video. But we had Erwin Chargaff. Now, he really didn't do anything specifically with the concept of DNA structure. What he did, though, was he concluded that in a sample of DNA, the amount of A was equal to the amount of T. Now, he didn't say anything about them pairing together. That conclusion was put together later by Watson and Crick. But he just said, percentage-wise, the amount of A was equal to the amount of T, and the amount of C was equal to the amount of G. And that was later helpful by Watson and Crick to determine that base pair rule. You definitely saw some Rosalind Franklin and Maurice Wilkins. In fact, there's a whole movie on the discovery of the DNA. We're really kind of supposed to watch it. I don't know that we're going to have time, but it really shows you kind of the essence of these people and their personalities. And what you find is that Rosalind Franklin, now she's a woman in science in the 1950s, so she was not considered an equal, and it was harder for her to do her job and be taken seriously. And then Maurice Wilkins was working in the same lab as Rosalind Franklin, and they both were doing a technique called X-ray diffraction. And this helped to give us this famous picture, as you saw in the video, this picture right here. Now, this picture was taken by Rosalind Franklin. Maurice Wilkins is actually the one that showed it to James Watson. But what you find in the movie about the discovery of the DNA structure is that Maurice Wilkins is kind of whiny. And he ended up getting the Nobel Prize. I don't know that he was deserving, but Rosalind Franklin had passed away by the time the Nobel Prize was awarded, not surprisingly by cancer, because they were working with X-rays. But essentially, it was this use of X-ray diffraction that created an image which led to the knowledge that DNA was a double helix. And then it was Watson and Crick who put it all together. They kind of took the work of Chargaff and the work of Wilkins and Franklin, and they were able to build the first model. Now, they did build one initially, and it was a giant joke, and they got it all wrong, but they eventually ended up getting it right, and so they were awarded the Nobel Prize. And so this model that they built is still the model that we know today, and it's hung around for a long time, so it must be working for us. But it was so simple, and yet it explained how information could be carried to offspring, and it also explained how changes could occur over time. And so what we're about to talk about now is this actual structure that they discovered. And so the structure of DNA, this is where your notes start. It is a polymer of nucleotides. This is a word that we should already be familiar with back from macromolecules. So the nucleotide is the monomer. It's the single unit that makes up the polymer. The polymer is going to be like the chain of nucleotides. And each nucleotide has three parts. We have deoxyribose is our sugar. It is a 5-carbon sugar. And then we have a phosphate group, which is just a phosphorus and some oxygens. And then we have one of 4 nitrogen bases. In DNA, that could be adenine, thymine, cytosine, or guanine. So just your basic nucleotide structure is a sugar, a phosphate, and a nitrogen base. Now the actual DNA polymer is a double helix. It has 2 strands of nucleotides. They are twisted to form a double helical structure. Now this is something that you need to understand, because this structure is what allows DNA to easily be copied and to be transferred from one generation to the next. This is what allows for DNA to be opened up and for genes to be copied for protein synthesis. And so the basic structure is, there's a lot in this one slide. Okay, so let's kind of work through it. So first off, the sides of the ladder are sugars and phosphates. Okay, we've got sugar, phosphate, sugar, phosphate, sugar, phosphate. The sugars are always our little pentagon structures. So the phosphates are usually represented by circles. But as you can see, the sides of this ladder are alternating sugars and phosphates. Okay. Now the inside of the ladder, or the steps, or the rungs, are our nitrogen bases that are held together. Now out here we learned that these have phosphodiester linkages. Now the sugars and the phosphates are held by a strong covalent bond, so the backbone of the molecule is strong. Now the inside, where those bases are held together, those are held by hydrogen bonds, and hopefully remember that hydrogen bonds are weak. And that's going to be significant, because even though there are millions of these bonds that are holding these bases together, we have to come in and break them open to copy this molecule. And so we would not want really, really strong bonds here in between these bases, otherwise we would never be able to open the molecule up. So we have strong covalent bonds that are holding the backbone together, sugar, phosphate, sugar, phosphate, sugar, phosphate. Something I need you to understand right now is that there is nothing special about the sugar phosphate backbone. Every student in the room, every human being on the planet, every animal, every plant, every fungus, every bacteria, all DNA, that sugar phosphate backbone is the same, okay, for all organisms. So there is nothing unique or special about the backbone. The uniqueness lies in the bases, in the order of those bases. So you can see that they're on the inside. Now just to kind of preface what we're going to talk about here in a minute, you see how there's 2 kind of geometric shapes here. We have a pentagon and a hexagon, and then over here we just have 1. Okay, so your bases can either be double ringed like we see here, or they can be single ringed. And so we're going to talk about that here in just a minute. And you can see that a double always pairs with a single. So another important concept, and this is going to be important when we get to replication, is that the strands are anti-parallel, which means they run in opposite directions. So where one is going up, the other is going down, and we'll take a look at that when we get to replication. All right, so those nitrogen bases, like I just mentioned, some of them are purines, and some of them are pyrimidines. And your purines are your double-ringed bases. These are the ones that had 2 of those geometric shapes. And purines include A and G. And then pyrimidines have only a single ring structure, and those include T and C. Now, one way to remember your pyrimidines is that you can see there's a Y in pyrimidines, and both of your bases have a Y in them that are pyrimidines, okay? So A and G, purines, T and C, pyrimidines. And a purine always pairs with a pyrimidine. Now the specific base pair rule, and this is something that students normally remember pretty well from freshman biology, is that A always pairs with T. I've heard people say the apple is on the tree, and then C always pairs with G, the car is in the garage. I don't care how you remember it. To me, A and T spell a word, C and G have the same shape, however you want to remember it, but that's a must know that A and T always go together and C and G always go together. Now, A and T always are held by 2 hydrogen bonds, so I remember AT2, and then C and G are always paired with 3 hydrogen bonds, and so CG3, and that's how I remember that. And once again, this is what makes us special, okay? It's the order of the base pairs. Where I may have AAC, you could have AAG. And that's what separates us from each other. That's what separates us from other species. All of our uniqueness is going to be in the nitrogen bases. So that sugar phosphate backbone is just for basic structure. It's the bases that actually hold our genetic code. And so here's an example of the base pairing. So here's cytosine. Okay, you can see that it only has 1 ring, and it's paired with guanine, which has 2. And there you see 3 hydrogen bonds holding them together. This was one of the puzzle pieces that Crick was able to figure out, or Watson, or the combination of the two of them, is with the help of Chargaff's rule, and they were given some insight about the molecular structure of these bases. They were able to see how the bases fit together on the inside of DNA. So over here we have a single-ringed thymine and a double-ringed adenine, and they're held by 2 hydrogen bonds. All right, so just a quick review before we get into replication. Remember that DNA can come in different forms. Now the structure of DNA is always the same. It's always that double helix with sugar phosphate backbone, nitrogen bases on the inside. But most of the time when the DNA is in the nucleus of the cell, it is found in the form of chromatin, which is condensed. I mean, it is packed. What you have to understand is that there's a lot of DNA in a little tiny nucleus. You have about 6 feet of DNA in one nucleus. And so it has to wrap around proteins in order to kind of fit inside of that nucleus. So it wraps around these proteins called histones, and those little units together are called a nucleosome. And even in the chromatin form, it is condensed, okay? But when we get ready for cell division, remember it further condenses into a super tight chromosome, okay? So most of the time we're in the chromatin form. When we copy our DNA, we're copying it in the form of chromatin. We don't see those chromosomes until prophase, when mitosis is starting. And so here you have the DNA double helix. It wraps around proteins called histones. This right here is what would be called a nucleosome. And you can see that it kind of tightly compacts, okay? So this is kind of what chromatin would look like, and then it super condenses to form the chromosome structure that we're familiar with. So as we get into replication, back when we covered the cell cycle, we know that the replication of DNA occurs in the S phase. That's the synthesis of DNA, and it's really important that that happens before a cell is copied. This process is important because if there are errors, it can lead to mutations, which can lead to problems potentially. And so the DNA must be copied. It's actually what we call semi-conservative. Every time we make a new DNA molecule, it's going to consist of 1 original strand and 1 old strand. And this was one of the things that Watson and Crick were able to, once they built the model, it was easy to see how this molecule might be replicated. Now it says that it helps reduce copy errors, and it really does, because it's always kind of keeping an original in the mix. So if you think about this in terms of like photocopies, like papers, like the notes you have in front of you, if every year your teachers make copies of a copy of a copy of a copy, and that goes on for years and years, what happens is over time those copies really decline, because every ink spot, every issue that occurs on the copy machine, and I'm sure you've seen some papers where maybe the copy machine puts a line on the paper, or sometimes it will spread certain words apart. And so what happens is if you're always making copies of copies of copies, then you're accumulating those errors over time. But if you always go back to your original copy, it doesn't mean that errors won't be made, but you will reduce the number of errors that you see on those paper copies. And so that's the way DNA replication works, is an old strand is always going to pair with a new strand, and that's what we call semi-conservative. So what we're going to do is essentially we're going to open up the molecule. We're going to build two new strands on the inside, and then the old and new go together over here, and the old and new go together over here. So some other models that were suggested were the conservative model, the dispersive model, but this is the model that we accept today, the semi-conservative. And so just a quick overview, the basic steps is that DNA is going to be separated into two strands. So essentially those hydrogen bonds are going to be broken and the DNA molecule is going to be opened up. Each of those old strands serves as a template. It's essentially the instructions for building the new strand. And then we're going to build complementary strands. Now this is important because the two strands in a DNA molecule are not identical. They are complementary. And that is based on the base pairs, where one strand has an A, the other strand has a T. Where one strand has a G, the other strand has a C. So we're going to build our new strands based on this complement system. And it really is pretty quick. It happens very quickly, and it's pretty accurate. This job is going to be accomplished by many enzymes. And this may be, depending on the resource that you're looking at, about 1 in a billion. Now you have to understand that there are billions of base pairs in our DNA, so that does mean that mistakes are going to happen, possibly every time we copy it. But considering the number of base pairs, this is really a pretty good statistic, okay? All right, so let's talk about enzymes. Now remember that enzymes are proteins that have a very specific job, and this is a big chemical reaction that is going to involve the work of many enzymes. In fact, there's more enzymes than just what we see here. There's different types of polymerase, there's different nucleases, and so what we need to know is just kind of the basic function of each one. So I kind of like to equate them with a simple word or group of words, just to kind of help understand. But remember, they are always very specific in how they work and what they do, okay? Also notice they end in ACE, okay? ACE is that common ending for our enzymes. So first we have helicase. Helicase is going to unzip the DNA. This is the one that is going to move along and break those hydrogen bonds. So helicase is the unzipper, okay? That's the word I like to use for helicase. All right, polymerase, okay, DNA polymerase. Now notice it's got the word polymer in it. It is actually going to build the new polymer, and it does that by adding new nucleotides. So what's going to happen is, based on that complement system, DNA polymerase is going to add new nucleotides to build the new strand. And so I usually just refer to polymerase as the builder. Okay, so helicase is the unzipper, polymerase is the builder. It's actually going to be adding the new nucleotides. Then ligase is going to be a gluer. Okay, ligase is going to join Okazaki fragments, which we'll talk about here in a minute. But essentially ligase is going to join fragments together and kind of make them one solid strand. So I usually call it the gluer. Nucleases, okay, nucleases, these are a type of repair. They're editors. They're going to remove incorrect bases. Primase builds primers, okay, primase builds primers. We're going to take a look at the process here in a second. And then topoisomerases, these are the ones I'm probably least familiar with, but they have to do with an untangling. So you think about, you have these long DNA strands, so they help to kind of untangle and keep that, make it a little easier to copy that DNA. So now let's take a look at the actual process, okay, and we'll kind of talk about where some of these enzymes come in at the important stages. So first of all, it's going to start at what we call origin of replication. It does not start at one end of the DNA, and this is where it's important to understand that the DNA runs in opposite directions. And so it's actually going to start somewhere in the middle of that DNA strand, and it's going to open up in what we call the replication fork. So it's always going to kind of form this V when that DNA opens up. So we're starting somewhere in the middle, and it creates this replication bubble, and then it's going to go in both directions, okay? Now this is where we get into the basic processes, and we're going to spend a minute or two on this slide because it's important that we understand the work of polymerase. So helicase was the enzyme that opened it up, okay? It unzipped the DNA molecule at that origin of replication. Now polymerase is our builder, okay? Now one thing you hopefully know is that enzymes are always picky, and polymerase is very picky, okay? So polymerase can only build in one direction, okay? Polymerase can also only add on to a strand that has already started. Polymerase cannot add on to nothing, okay? Now when it comes to these directions, 5' and 3', I don't really know where the prime comes from, but I will tell you where the 5 and the 3 comes from. So back when we did glycolysis, we learned that in organic chemistry, the carbons are numbered, okay? So remember that deoxyribose is a 5-carbon sugar. And so here we have a number 1 carbon here. There's a number 2 carbon here. So there's 1. I don't like this thing. Here's my 3 carbon. Here's my 4 carbon. And up here is a 5th carbon, okay? So when we talk about 3 to 5 and 5 to 3, okay, what I need you to see is that when those sugars, the pentagons are pointed up, my 5 carbon is on top and my 3 carbon is on bottom. Okay, so this is 5 to 3. Now remember that the strands are anti-parallel, okay? So over here, notice the pentagons are pointed down, and so my 3' is up and my 5' is down. All right, when we get to the essays for this unit, you're going to be labeling diagrams, and it's really important that we understand this 5-3 concept, okay? So once again, the 5-3 goes back to the carbons. So because polymerase can only build in one direction, what that means is the new strands are going to get built in opposite directions, okay? So on one side, the DNA is going to build the strand in one long continuous strand. And this is going to be built toward the replication fork, okay? So I want you to kind of think of it like a zipper really quickly. Imagine if you had your jacket zipped up, okay? Now, as you unzipped, in the case of directions here, you're unzipping going down, right? Well, on one side, you could literally just build following that unzipping, okay? So as you unzipped, that DNA polymerase could just build, 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 and as you unzipped, it could just follow that replication fork, okay? Now, if on one side, the polymerase was building down and following that zipper, that means the other side, it's going to have to be built going up, going the opposite direction. And so if you imagine that zipper, when you unzip it a little bit, you can only go up for a short stretch. And then you'd have to unzip it some more before you could build another chunk. And then you'd have to unzip it some more before you could build another chunk. And so this is what happens with DNA polymerase. On one side, it builds one long continuous strand, and we call that the leading strand. I'm going to show you a picture here in a second. On the other side, it has to kind of go against the opening, and so it ends up building that strand in pieces called Okazaki fragments. And so we call that the lagging strand. And this is where that ligase is going to come in that we mentioned earlier, the glue. So this is all because polymerase is picky, okay? So right here is my replication fork, all right? You can see that as I'm unzipping, okay, I'm unzipping in this direction. As I'm unzipping, this leading strand is built, 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 and as that thing opens, we can continue to build. But on the other side, as it opens, I can only build little pieces at a time. So this is what we call our lagging strand. Now, another important thing to understand about polymerase is that polymerase has to have something to add on to, okay? Polymerase cannot add a nucleotide onto nothing. And so before polymerase can even build the strands of DNA, a primer has to be put in, okay? So DNA polymerase is not able to add DNA nucleotides without a primer. And so a primer is just a little short sequence of RNA that gets the strand started, okay? And that is going to be accomplished by the enzyme primase. So it's actually joining RNA nucleotides. We're going to talk more about RNA later. But RNA, it has a U instead of a T. It also has ribose instead of deoxyribose sugar. So there's definitely some differences. And so the leading strand is going to require one primer. And then it can build until it finishes. But the lagging strand is going to require multiple primers because every time a fragment is built, it's going to have to be started by a primer. Now, before we actually get our final DNA molecule, these primers are going to be cut out by nucleases, and they are going to be replaced by DNA nucleotides, and then ligase will come in and join it all together. Now, there's a good video for replication. I'm not sure if this link is it or not, but if not, I'm sure the video is going to be put on Google Classroom for you. But I would encourage you to watch that. It's really a great visual of the process of replication. Now, as I mentioned earlier, it's really a pretty efficient, accurate process. Polymerase is one of our editors. It kind of proofreads as it goes. But if it does make an error, okay, it could be what we call a mismatch error. And this is where you have like a paired with a C or something like that. So we have a whole group of enzymes that can go in and kind of repair and cut out some of these mismatches. So those nucleases that we mentioned, they're able to cut out damaged pieces, and then polymerase will come in and fill in the gap. Now, even with all of our proofreading and repair, mistakes can still happen just because of the sheer number of nitrogen bases. Now, the last thing we're going to talk about is telomeres. And so this is, in order to understand this, we kind of have to understand that leading and lagging strand. So remember, polymerase cannot add on to nothing. And so at the end of our chromosomes, we have these long repeat sequences. In fact, a lot of our DNA are regulatory sequences. They aren't actually gene coding. But every time we copy our DNA, our chromosomes get a little bit shorter. Okay, and the reason why is because on that lagging strand, remember, DNA polymerase cannot add on to nothing. And so once you remove that initial primer, it can't be replaced, okay? Because the DNA polymerase has to have something to build off of. So that initial primer that's put in, it can't be replaced, which means every time the DNA gets copied, it gets a little bit shorter, a little bit shorter, a little bit shorter. And so to kind of help protect our genetic information, on the ends of our chromosomes, we have these long repeat units, okay? It could be 100 to 1,000 of these repeats of this particular sequence usually. And that way, every time it does shorten, we're not losing critical genetic information. Now, there is an enzyme called telomerase that actually restores those telomeres. And this is one of the things that can be studied in terms of aging. This is one of the things that can be studied in terms of cancer, because a lot of cells don't have telomerase, which means as we get older and that cell is copied more and more and more and more, then eventually we may get past those telomeres and get into critical genetic information. And so that could lead to some cells that eventually become no good. They could eventually become cancerous, or that could lead to just the basic aging process. And so it seems, though, that cancer cells oftentimes have this telomerase, which allows them to kind of keep going, because normal cells will eventually lose critical DNA and they'll maybe go through apoptosis or they will die, whereas those cancer cells don't. So it could be that they have telomerase where some of our normal cells don't. And so this just kind of shows you how we get that. So here's a primer that after the primers are replaced, see, this one can be replaced because there's something to build off of, but this one here is unable to be rebuilt, okay? Same thing with this one here. And so over time, what happens is we get the ends are not going to be fully copied. And so they get a little bit shorter and a little bit shorter and a little bit shorter. But luckily, those telomeres help give us some extra cushion so that we're not deleting critical genetic information. All right, so to wrap it up, okay, once we get all of our strands copied, we now have 2 identical molecules of DNA. Remember that an old strand is always going to pair with a new strand. That's the semi-conservative. And then the new strands are going to recoil, and we will have 2 identical DNA molecules. It looks something like this. And so when it's all said and done, we have our new molecules of DNA that are then going to be separated through the process of mitosis, and those new cells will continue on. Now, this is not something you have to know, okay? This is not something you have to know at all. This is just extra information. But I mentioned earlier that there's about 6 feet of DNA in each cell, or 2 meters of DNA. We have about 75 trillion cells in the human body. And so if we were to take all of our DNA and stretch it out, we could go from the Earth to the Sun more than 400 times. And this diameter, so you may think, well, geez, that's a lot. 6 feet is a lot, but DNA is still a tiny molecule. Normally I would kind of get a meter stick and kind of try to get you to understand this here. But this is the diameter of DNA, okay? It's like 2 billionths. It's 2 billionths of a meter. And so imagine if you took like a millimeter and cut it into like a million pieces. 2 of those would be the diameter of DNA. So you can still see that it's a really tiny molecule. Like I said, you don't have to know this for the test. It's just kind of an FYI. So tomorrow we're going to be doing an activity to kind of help review DNA replication. Just be sure you understand the structure of DNA and how it's copied. And hopefully I will talk to you soon. Y'all have a great day, and I'll see you later.